Hello students, welcome back to my channel. In this session, we are going to learn about cell division. What is cell division? Cell division, it is a biological process by which a parent cell divides to form two daughter cells. So this process is essential for growth, development, reproduction and tissue repair in all living organisms. And there are two types of cell division, that is mitosis and meiosis. So mitosis, it is responsible for producing new body cells as well as repair of injured cells. So these are the few examples for mitosis. Here the parent cell will be divided into two new daughter cells. And here repair of injured cells, that takes place in starfish, we call it as regeneration. But the process here is mitosis. And the vegetative type of reproduction of plants also comes under mitosis. And the name will be different. Formation of two new daughter cells from a parent cell in amoeba, we call it as binary fission, and in hydra we call it as budding, but the procedure is same. The next type of cell division is meiosis. And meiosis takes place only in reproductive cells because an individual has 46 chromosomes in 23 pairs. If this same number of chromosome is present in egg as well as in the sperm, we will get more number of chromosomes, which will not be suitable for the individual. To reduce that, the egg and the sperm will have the half the number of chromosomes, that we call as haploid number of chromosomes, which fuse together to form the diploid number of chromosomes. Okay, so here the reduction of a diploid into a haploid cell, we call it as meiosis. And this also we can see in plants. So the sperm and the egg will be present in both male and female flower, and it contains the haploid number of chromosomes in egg as well as in the sperm, and after fertilization it becomes a zygote which is in diploid format and it will be developed into a new individual plant. So conversion of a diploid cell into a haploid cell, we call it as meiosis, and this takes place only in the reproductive part of an organisms. First, we can learn about mitosis. So, in mitosis, there are various stages, which we call as, the first stage we call as interphase, and then prophase, prometaphase, metaphase, anaphase, telophase, and then cytokinins. Okay. Now, what happens in each stages? So, in mitosis, interphase is an important stage where the cell prepares itself for the division. So in the interphase, it is divided into G1 phase, S phase and G2 phase. So in G1 phase, the cell is getting ready for the division, and in S phase, the DNA present in the chromosome, it gets duplicated, that is copied. So exact copy of a parent DNA is copied into another one new DNA. And in G2 phase, it is now fully prepared for the mitosis division. So what happens in mitosis? The first stage is prophase, next one prometaphase, metaphase, anaphase, telophase, and then cytokinins, and then it divides into two individual cells. Okay, this is the outline of the mitosis, what happens in the mitosis. Next, we will learn one by one stage in detail. So the first stage is prophase. So what happens in this prophase stage is, the chromatin fibers, they condense into chromosomes, and they are seen clearly, and two identical sister chromatids we can see with the centromere, and the mitotic spindle begins to appear. So it looks like this prophase. So you can see the micro spindle fibers appearing at both the ends of the cell, and then you can clearly see the chromosomes with the centromeres. See, and this two we call as the chromatids, and they can be clearly seen. They condense into chromosomes. So under the microscope, we can see like this prophase. So in a cell, this is clearly visible in onion root tip. If you see under the microscope, see, this is the cell and the chromosomes contains in the center of the cell. So it is ready to what? Cell division, that is, it is the DNA is copied and the microspindles begins to appear at the both the ends of the cell. So it is clearly visible. That is we call as the prophase. That's the stage 1 phase. And the next one is metaphase. So what happens in the metaphase? Now the chromosome is present in the center of the cell. And then here in metaphase, it lines up in the middle of the cell, in the equatorial plane. It lines in the equatorial plane and it get attached to the microtubules. So in the prophase, we learned that we have micro spindles at both ends of the cell. No? So it gets attached to that microtubules. So can you see in this picture? See the chromosomes, they get arranged in the equatorial plane, that is the middle of the cell, and the centromere will be attached to the microtubules at both the ends of a cell. So this stage we call it as metaphase. And next stage is anaphase. So what happens here? Since it is attached with the microtubules, now the microtubules pushes the chromosomes to both the ends. So the sister chromatids, they get break and the chromosomes move to the opposite ends. See, you can see clearly in the picture. So in the metaphase, it gets arranged in the center of the cell in equatorial plane, and in anaphase, the sister chromatids break and each one moves to the opposite ends of the cell. So this stage we call as anaphase. So under the microscope, we can clearly see that inside the cell, you can see the sister chromatids, they are breaking and moving to the both the ends of a cell. Okay. And the next stage, that is telophase, it is the last stage. Now the chromosomes has end to the both the sides of the cell. And now what happens? Nuclear envelope begins to reappear. What is the nuclear envelope? It is the covering of the nucleus. No, so it begins to develop, and the microtubules which is holding the chromosomes, they begin to disappear. You can see this clearly in this stage. See telophase, that is now the chromosomes are moved to the opposite side, and the nuclear envelope, it begins to develop around the nucleus, and the microtubules, it begins to disappear. Okay. So you can now see a partition inside a cell into two different cells. It is dividing into two different cells. You can see this clearly. This stage we call it as telophase. And what do you mean by cytokinesis? So along with the telophase, what happens? The cytoplasm and the other cell organelles also, they begin to move towards both the ends of the cell. And then you can see a contractile ring at the center of a cell. So it forms as a cleavage, for that is it splits the cell into two new daughter cells with chromosomes inside and a fully developed nuclear envelope. Do you understand this? Yes. So we learned about mitosis. So what are the stages in mitosis? Interphase, prophase, prometaphase, metaphase, anaphase, and telophase. So what happens in the interphase? The cell prepares itself for the growth, and the major thing happening in interphase is DNA is replicated. Okay, the cell prepares it for mitosis. And what happens in the prophase? That we learned. The chromosomes can be seen visible in this stage. And the metaphase, it arranges in the center of the cell. And in anaphase, what happens? The microtubules, it pushes the chromosomes to two opposite ends of the cell. And in the telophase, the cells the chromosome reaches the opposite sides of the cells, and the two nuclei begins to form, and the parent cell will be divided into two new daughter cells. So this is known as mitosis, and it helps in cell growth and repair of the cell in living organisms. So these are the few examples of mitosis. In first also you saw some examples. No, these are the examples in plants as well as in animals. Okay, so in bacteria, that is prokaryote. So here this process known as binary fission. Amoeba, this also known as binary fission. And in hydra, we call it as what? Budding. And yeast also. So the name will be different, but the process is what? Mitosis. So regeneration. So if you cut a small thing from the starfish, it develops into an individual one. That is known as the process known as cleavage. But what happens there? Mitosis takes place. It helps in the cell growth. I hope you understood about this mitosis. Yes. Next, we can learn about meiosis. What is the difference between meiosis and mitosis? Because both comes under cell division only. So what is the difference between these two? We can learn now. Meiosis. What is meiosis? Meiosis, it is used for the production of gametes, that are sex cells. That means we can see only in the reproductive cells. This cell division takes place only in a reproductive organ. Okay. So in that, the diploid cell will be converted into haploid cell, and again after fusion the haploid cell will form a what diploid cell, that we call as, and we develop into a new genome. So this process takes place in meiosis. I can see here the stages in meiosis of two types, that is meiosis 1 and meiosis 2. In meiosis 1 we have the same name: interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and again the cell undergoes again what prophase 2, metaphase 2, anaphase 2, telophase 2 and cytokinase 2. That is second division, which develop into 4 haploid cells. I told no, the diploid cell will be converted into 4 haploid cells. Okay, so what is happening? So here in meiosis 1, what happens? The prophase, the same thing, the chromosomes get condensed in the center of the cell, but here something happens. Okay, and then the other procedures are same. Metaphase, it get arranged. Anaphase, it is separated the both the ends, and in telophase the parent cell will be divided into two equal what daughter cells, and again the daughter cells, the two haploid cells, again it undergoes meiosis to the same thing, the prophase, metaphase, anaphase, telophase, and again it forms what 4 haploid cells. So let us learn what happens in the meiosis 1 stage. What is the difference in that? So in meiosis 1, what happens means the DNA is broken at same spot on each homolog chromosomes. The two chromosomes we call as homologous chromosomes, and then what happens? A crossing over takes place with the help of a protein structure called synaptomal complex, and the junction between two DNA we call it as chiasmata. That is a cross-shaped structure we can see there. So the exchange of gene takes place exactly in that place. So you can see clearly crossing over. So we say this as homologous chromosomes. Okay, the same chromosomes. See here in prophase, what happens? Exchange of chromosomes takes place. See, this is known as crossing over, and the point at which the two chromosomes cross each other we call as chiasma. Okay, crossing over takes place, and here you can clearly see recombinant chromatin. So gene from here is interchanged to the other gene, and from here some piece of DNA is transported to the. Why does this happens? Because so by crossing over it results in genetic diversity. We have diversity of species. Okay, do you understand about that? So, in summary, we can see that mitosis, it is responsible for the cell division, and it is very important because each daughter cells, it carries the genetic material from the parent, and it is essential for maintaining health and function of an organisms. And coming to the meiosis, so the diploid number of chromosomes, it get reduced to haploid number of chromosomes, and this we can visualize only in the reproductive cells. So once it get fused, that means fertilized, it becomes an diploid cell and it develops into a new individual cells. To reduce the chromosomes into a half number, the meiosis takes place in the reproductive cells. And the stages are same. In mitosis, we have prophase, metaphase, anaphase, and telophase. As such, we have the same phase: prophase, metaphase, anaphase, telophase in meiosis also. But in prophase 1, crossing over takes place. You have to keep that in mind. Crossing over is nothing but exchange of genes between two chromosomes. It leads to genetic diversity of living organisms on the world. Hope you all understood this chapter. Thank you. Thank you for your listening and happy learning.